We know that cells harvest energy from food. They do this using two primary metabolic pathways, called glycolysis and the citric acid cycle. These two pathways generate the NADH necessary to feed electrons to the electron transport chain. Let's use this opportunity to explore these two pathways in greater depth and to see how they generate NADH from food. Glycolysis is a 10-step pathway in which one molecule of glucose is converted into two molecules of pyruvate and two molecules of NADH. The first step is the phosphorylation of glucose to form glucose 6-phosphate. This reaction is catalyzed by the enzyme hexokinase and also consumes one molecule of ATP, which we have omitted from this image for clarity. In the second step, glucose 6-phosphate is isomerized into fructose 6-phosphate, as catalyzed by the enzyme phosphohexose isomerase. In the third step, fructose 6-phosphate is phosphorylated to produce fructose 1,6-bisphosphate, as catalyzed by phosphofructokinase. At this stage, the cell has already invested two ATP molecules, but it will generate many more in the later steps of this process. In the fourth step, fructose 1,6-bisphosphate is cleaved into two 3-carbon molecules called glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. We started with a single molecule of glucose, a 6-carbon sugar, and now we have two different 3-carbon sugars. Glycolysis proceeds with only one of them, the glyceraldehyde 3-phosphate. Thus, in the fifth step of glycolysis, dihydroxyacetone phosphate is also converted into glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. During the sixth step, glyceraldehyde 3-phosphate is oxidized to form 1,3-bisphosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase. The reaction is coupled with the reduction of NAD+ into NADH. Since the initial glucose molecule was split into two 3-carbon sugars, for each glucose molecule that enters glycolysis, two molecules of 1,3-bisphosphoglycerate and two molecules of NADH are produced. In the seventh step, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate by phosphoglycerate kinase, generating ATP in the process. Again, since there were two 3-carbon sugars generated from every glucose, this step produces two ATP molecules for every glucose molecule consumed. In the eighth step, 3-phosphoglycerate is converted into 2-phosphoglycerate by phosphoglyceromutase. In the ninth step, phosphoenol pyruvate is formed from 2-phosphoglycerate by enolase. And in the last and 10th step of glycolysis, pyruvate kinase converts phosphoenol pyruvate into pyruvate, also generating ATP in the process. Again, this step produces two ATP molecules for every glucose molecule consumed. So now that we have been through the 10 steps of glycolysis, where are we with the number of ATPs and NADHs that have been produced? Remember, one molecule of ATP per glucose is consumed in each of steps 1 and 3. In steps 7 and 10, the cell capitalizes on this initial ATP investment by generating two ATP molecules per glucose. This means that 4 total ATPs are made. This gives us 2 net ATPs per glucose at the end of glycolysis, and even more importantly, 2 molecules of NADH. At this point, glucose has been partially oxidized to pyruvate. It is now up to the citric acid cycle to complete the oxidation of pyruvate to carbon dioxide.
So let's give ourselves an overview of glycolysis. And glycolysis is an incredibly important biochemical pathway. It occurs in practically all life as we know it. And it's all about taking glucose as a fuel, and in the process of breaking it up, lysing the glucose, glycolysis, breaking it up into two pyruvate molecules. Glucose is a 6-carbon molecule. Each of the pyruvates are 3-carbon molecules. In the process of doing that, you produce 2 ATPs net. It actually turns out that you need to use 2 ATPs, and then you produce 4. So you use 2 ATPs. That's often called the investment phase, and we'll talk about that in a second. And then you produce 4 ATPs for a net of plus 2 ATPs. And that's what we see right over here. You see a net of 2 ATPs being produced directly by glycolysis. And then you also have the reduction of NAD to NADH. Remember, reduction is all about gaining electrons. And over here, NAD, that's nicotinamide adenine dinucleotide. We have other videos on that. It's an interesting molecule. It's actually a decent sized molecule. We see this positive charge, but then we see that not only does it gain a hydrogen, but it loses its positive charge. So it gains a hydrogen and an electron. You could think on a net basis, it's gaining a hydride. It's gaining a hydride. Now, a hydride anion is not going to typically be all by itself, but on a net basis, you can think about that's what's happening. And so it's gaining a hydrogen and an extra electron. And so the NAD+, this is going to get reduced. That is going to get reduced to NADH. So this is getting reduced to NADH. And that NADH, once it can then be oxidized in the electron transport chain. And we'll study that later on when we think about oxidative phosphorylation to produce even more ATPs. But on a very high level, simple basis, glucose being broken down in pyruvate, 6 carbons, 3 carbons each of these pyruvates. Now there's other things attached to the carbons, and we'll see that in a little bit. 2 ATPs net generated. And you have the reduction of NAD, 2 NADs to 2 NADHs, and those can be used later on to produce more ATPs. Now, glycolysis is typically just the beginning of cellular respiration. If oxygen is around, then you have these products, some of these products moving into the mitochondria, where you can have the citric acid cycle, Krebs cycle, and the oxidative phosphorylation occur. If you don't have oxygen around, then you're going to do anaerobic respiration, or you're going to go into fermentation. We'll talk about that in a future video. And that's really about figuring out what to do with these products, and especially replenishing your NAD+. Now that we have a very high-level overview of glycolysis, let's get a better appreciation for exactly what's going on. And whenever I look at these more detailed processes, the one thing to just appreciate is how much complexity is occurring in all of your cells right now. This isn't it is fairly abstract to just even imagine these things. But this is happening throughout your body gazillions of times right now. This isn't something that's somehow distant from you. And it's also fun to appreciate, well, how all of this was discovered by scientists. That's a whole other fascinating discussion. But the whole point of this video is just to give us an appreciation for the actual mechanism or the reaction by which it occurs. I'm not going to go into the detailed organic chemistry mechanism. So over here, this is a glucose molecule over here. You see 1, 2, 3, 4, 5, 6 carbons. And then the first step is it gets phosphorylated. And we have a whole video on the phosphorylation of glucose. And all of these steps are facilitated with enzymes. The phosphorylation is facilitated with the hexokinase. Kinase is a general term for an enzyme that either facilitates phosphorylation or dephosphorylates. It's dealing with phosphorylation, I guess you can say. And all enzymes are all about lowering the activation energy. And the way that hexokinases do, or part of how they do it, is they involve the cofactor magnesium ion. And we've talked about that in other videos, how cofactors can help an enzyme lower the activation energy. And to do the phosphorylation, we use an ATP. So this is -1 ATP. So we are in the investment phase. We are in the investment phase right over here. But this reaction, it strongly goes from left to right. It's a coupled reaction that phosphorylating the glucose, that requires free energy, but the ATP releases free energy. You couple these reactions, and it strongly goes from left to right. Now, and just to be clear, what happened? This over here got replaced, or maybe I should say this over here got replaced with that over there, just to keep track of what's happening. Now, another enzyme catalyzed reaction. This one is actually in equilibrium. It can go both ways. But as we'll see, the right side, or the things that are further into the glycolysis process, these are constantly being turned into further products, so their concentrations are going to go down, and so the reaction will tend to go that way. Although this particular reaction, going from glucose 6-phosphate to fructose 6-phosphate, this could be in equilibrium. But the enzyme that facilitates this, phosphoglucose isomerases, these are enzymes that help go from one isomer of a molecule to another isomer. And that's what's happening here. Instead of this oxygen being bound to this carbon, this bond forms with this carbon. And so you have fructose. You have the 5-carbon ring over here, or the 5-element ring. You have 4 carbons in it, versus a 6-element ring, where right over here you have 5 carbons. So this bond goes to this carbon right over here, and that's the main difference. And then you have another very strong forward reaction, once again facilitated by ATP, and this is done by phosphofructokinase. It has the word kinase in it, and it's using up the ATP. You can guess what's going to happen. We're going to attach another phosphate group to the fructose 6-phosphate, and now you have 2 of these phosphate groups. So this hydrogen right over here is now replaced with another phosphate group. And once again, it's facilitated by the magnesium cofactor. It helps stabilize some of the negative charge associated with the phosphate groups. We talk about that in other videos. But the important thing is it uses another ATP. We're still in the investment phase. -1 ATP. And every time I look, it's just fascinating that all of this stuff is happening in your cells as we speak. In fact, in order for me to speak, this has to happen, because my body needs to take glucose and come up with some energy to turn to ATPs so that my muscles can actually move and I can actually inhale and exhale and all the things that I need to do for speech. So appreciate what's going on over here. Now, the next step we talk about, the whole process of glycolysis is lysing glucose. And over here, this is derived from glucose and some phosphates. And the next step, we're actually going to break it up. And we're going to break it up using the enzyme fructose biphosphate aldolase. Aldolase enzymes, they facilitate the aldol reaction. And in this one, we're going to actually, the aldol reaction could be to merge to put together 2 molecules, or in this case we're going to break them up, and we break them up into 2 3-carbon chains. Now these 2 3-carbon chains, glyceraldehyde 3-phosphate and this character right over here, they can be converted between the 2 with another isomerase, this triose phosphate isomerase right over here. So at this point in glycolysis, we can think of ourselves as really having 2 of these. So let's say 2 times glyceraldehyde 3-phosphate. So as we go further on, just imagining this happening twice for every glucose molecule. And at any time you get confused, I encourage you to pause the video and see how these pieces and these pieces put together can form that over there. So now we have another reaction. It's facilitated by a dehydrogenase. Dehydrogenases usually are involved in this case. This is the reduction of NAD. We saw that in the overview video. So NAD is being reduced, and this can be used, this NADH later on can be used in the electron transport chain to potentially produce some more ATP. But in that process, we also add another phosphate group to the glyceraldehyde 3-phosphate. So you see this phosphate group, right over here that wasn't there before. And actually, this right over here is, I should have arrows on both sides. This right over here, that reaction could actually go both directions. Actually, that reaction can as well. And then we are now going to be in the payoff phase. So this right over here, we're starting with this molecule that has these 2 phosphate groups. And then using the phosphoglycerate kinase, we're able to pop one of those phosphate groups off and in the process produce ATP. Now we might want to say +1 ATP, but we have to remember this is now happening twice because we had 2 of those glyceraldehyde 3-phosphates. So now we could say, if we're talking about this happening twice, 2 + 2 ATPs, we are now in the payoff phase. Then you have, facilitated by the phosphoglycerate mutase. A mutase is a class of isomerases, I have trouble saying that, that'll take a functional group from one place to another, or take one part of a molecule to another part, and you see this phosphate group. You see this phosphate group moving on from this carbon to the middle carbon. And so that's what that's doing. Then we use an enolase to get over here, and then the pyruvate kinase. The pyruvate kinase, and here the kinase is going to be used to dephosphorylate this molecule right over here, and it gets us to, the way I've drawn it is pyruvic acid, since I have the hydrogen here, and if the hydrogen is let go and this oxygen hogs the electron, we would call this pyruvate, and this is considered to be the end of, I guess you could say, mainstream glycolysis. But what happened, and I don't want to glaze over what happened over here, this ADP got converted to another ATP, but it's going to happen twice. So this is another +2 ATPs. So hopefully you see the investment phase, we use an ATP right over here to phosphorylate the glucose. We use another ATP right over here to throw that second phosphate group on the fructose 6-phosphate. But then we get the payoff phase. So we're able to produce this NADH, and this is actually going to be 2 NADHs because everything here is going to happen twice now. We can assume that this character over here also gets converted to a glyceraldehyde 3-phosphate. And now we produce 2 ATPs because this is happening twice, and we produce 2 ATPs right over there. So hopefully everything we talked about in the beginning, everything we talked about in the beginning, actually makes sense.
In this video, we will study about the process of glycolysis. The word glycolysis is made up of 2 words. The word glyco means glucose and the word lysis means breakdown. The glycolysis is a metabolic pathway that involves systematic breakdown of glucose to produce energy. And the glycolysis takes place in the cytosol of the cells. First, we will have an overview about this process, and then we will study about the various reactions involved. In glycolysis, one molecule of glucose, which is a 6-carbon compound, is broken down into 2 molecules of pyruvate, which is a 3-carbon compound. And in this conversion, energy is released in the form of ATP and NADH. The pyruvate formed is further utilized in various energy producing pathways that produces more energy. The process of glycolysis takes place in 2 phases. In the first phase, the glucose is first converted into 2 molecules of glyceraldehyde 3-phosphate. Since this conversion involves addition of phosphate, the first phase actually uses energy instead of producing it. In the second phase, the glyceraldehyde 3-phosphate is converted into pyruvate. And it is in the second phase energy is actually produced. Now let's look at the various reactions involved in the glycolysis. So it all begins with a molecule of glucose that enters the cytosol. And the first reaction that takes place is conversion of glucose to glucose 6-phosphate. The enzyme that catalyzes this reaction is called hexokinase. The word hexo means 6, denoting the 6-carbon compound glucose, and the kinases are a group of enzymes that catalyze the phosphorylation reactions. So the enzyme hexokinase transfers a molecule of phosphate from ATP to glucose and converts glucose to glucose 6-phosphate. In this reaction, ATP is converted into a molecule of ADP. Hence, it utilizes 1 molecule of ATP. Next, the glucose 6-phosphate is converted into its isomer, the fructose 6-phosphate, by the enzyme isomerase. This is a simple rearrangement reaction. In the next step, the fructose 6-phosphate is converted to fructose 1,6-bisphosphate. That is addition of another phosphate group at the first carbon of the fructose ring. This reaction is catalyzed by the enzyme phosphofructokinase, another kinase enzyme which transfers a group of phosphate from ATP to fructose 6-phosphate, and again ATP is converted into ADP, hence another ATP is utilized in the reaction. Next, the fructose 1,6-bisphosphate is split into 2 molecules, the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These both are 3-carbon compounds, and the enzyme that catalyzes this reaction is called aldolase. These all reactions compose the first phase of the glycolysis, And as you can see, energy is actually utilized in this process in the form of 2 ATPs. The two compounds glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are actually isomers and can be interconverted into each other by the enzyme isomerase. But since the glyceraldehyde 3-phosphate is further utilized in the process of glycolysis, the equilibrium of this isomerization reaction is always towards the glyceraldehyde 3-phosphate. Also keep in mind that every molecule of glucose is split into 2 molecules, so every reaction of the second phase happens twice and produces twice the products. In the next reaction, the glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. This involves addition of a phosphate group at the first position of the phosphoglycerate. The enzyme that catalyzes this reaction is the glyceraldehyde 3-phosphate dehydrogenase. And in this reaction, nicotinamide adenine dinucleotide, which is the coenzyme, is reduced to NADH, which is further utilized in the electron transport chain to produce more energy. Also, this reaction utilizes a molecule of inorganic phosphate. The 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. This kinase enzyme transfers the phosphate group at the first position of phosphoglycerate to adenosine diphosphate, and in this process a molecule of ATP is generated. Next, the 3-phosphoglycerate is converted to 2-phosphoglycerate. This is a simple rearrangement reaction that happens with the help of the enzyme phosphoglycerate mutase. Next, the 2-phosphoglycerate is converted to phosphoenol pyruvate. The enzyme involved is enolase, and magnesium ions are also utilized in this reaction. In the final step of the glycolysis, the phosphoenol pyruvate is converted to pyruvate by the enzyme pyruvate kinase. The phosphate group at the phosphoenol pyruvate is transferred to ADP and converted to ATP. This all composes the second phase of the glycolysis. And as you can see, energy is produced in the second phase of the glycolysis. Now let's first look at the net reaction of glycolysis and then we will understand it in brief. We have one molecule of 6-carbon glucose that splits into 2 molecules of 3-carbon compound pyruvate. There is a net utilization of only 2 ADPs. Since 4 ADPs are used in the second phase, but 2 ADPs are also produced in the first phase, there is a net use of only 2 ADPs. 2 molecules of inorganic phosphate are utilized in this step. 2 nicotinamide adenine dinucleotides are reduced to NADH molecules in conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate.
Welcome to our video series on cellular respiration. Before we get started, I'm going to ask you to do a few things. Don't be a passive learner. Have paper and pencil handy. Pause the video frequently. Rewind and rewatch. Draw your own pictures, because as you know, pictures are your friends. And ask questions. Go ahead, write them down. Research the answers. And to really test your understanding, teach it to someone else in your own words. And if you want, you can leave questions in the comment sections below, and I'll answer them as soon as I can. Now let's get started. I decided to make this a 3-part series. In part 1, we'll do an introduction to cell respiration and the details of glycolysis. That's what you're watching now. Then in part 2, we'll take a closer look at the Krebs cycle. And in part 3, we'll look at electron transport phosphorylation. Watch these 3 videos in sequence. I'll have links for these second 2 at the end of this video. And then after you're finished with that series, check out my video on fermentation. We'll look at a different way of getting the energy out of glucose. Alright, in our last video series, we looked at photosynthesis, how we could take the energy of sunlight and put it into the bonds of glucose to make our cellular fuel. So what we need to do in this video is figure out how we get this energy back out. We need to turn photosynthesis around so we can see respiration. We're going to get the energy back out of glucose in a form called ATP. So when we look at this overall equation, we're taking glucose in the presence of oxygen, and we're going to break it apart into carbon dioxide and water to liberate energy in the form of molecule ATP, approximately 36 of them. Cellular respiration is going to occur in 3 stages. The first stage is called glycolysis. It's an anaerobic stage that takes place in the cytoplasm. The products of glycolysis will enter the mitochondria, where the second and third stages of respiration occur. The second stage of respiration is called the Krebs cycle. It's preceded by a preparatory step, and is a series of reactions that occur in the mitochondria that are often referred to as the citric acid cycle, because the first intermediate made is citrate or citric acid. And the final stage of respiration is electron transport phosphorylation. It takes place deep inside the mitochondria. It's a series of enzymes embedded in the inner membrane called the electron transport chain. It's this stage of respiration that's aerobic and that will produce the majority of our ATP. Now let's take a look at where this occurs. Here we have a cell, and we want to zoom in on some important organelles. The mitochondria. There we go, the mitochondria. Let's take a closer look at these mitochondria. The mitochondria is often referred to as a cell's power plant. It's the primary site of cellular respiration, where we're going to recapture the energy from those high energy bonds of glucose. And you'll notice that the mitochondria is a double membrane bound organelle, and it's across this inner membrane where a lot of our actions are going to occur. We'll come back to the mitochondria later, because the first stages of respiration, glycolysis, occurs out here in the cytoplasm. Now let's keep in mind what it is we're trying to do. We're trying to make ATP. So we need to talk about how ATP is actually made. It turns out there are 2 ways to make ATP: substrate level phosphorylation and electron transport phosphorylation, which is driven by chemiosmosis. Now let's look at what substrate level phosphorylation looks like. In substrate level phosphorylation, a substrate directly hands an ADP, a phosphate, to become ATP. Let's watch that again. We have substrate level phosphorylation. ADP was phosphorylated into ATP with a direct transfer of a phosphate group. So we call this substrate level phosphorylation. The substrate donates the phosphate. In electron transport phosphorylation, production is driven by a chemiosmotic gradient, the flow of protons through a special channel protein that we call ATP synthase. So let's get started with glycolysis. Remember that glycolysis is anaerobic and it occurs in the cytoplasm. And we're going to start with glucose. And in the first step of glycolysis, glucose is split with the energy from ATP. And this is interesting because we're trying to make ATP, but in the first step, we actually consume 2 ATP to split glucose into 2 molecules called PGAL, or phosphoglyceraldehyde. So in our first step, we take a 6-carbon molecule and split it into 2 3-carbon PGALs, and it's going to cost us 2 ATP. We call this our energy investment stage. We spend 2 ATP to make 2 PGAL. Now these 2 PGAL molecules are used to reduce NAD+ into NADH, and in doing so, the PGAL are converted into BPGA, or biphosphoglycerate. Now these 2 molecules of NADH are carrying for us high energy electrons. They're very similar to the electron carriers NADPH that we saw in photosynthesis, but obviously minus a phosphate, so it's NADH. But these 2 molecules have a lot of energy left in them. They're carrying this high energy electrons, and that's energy we can tap into later. Let's look at it again. The 2 PGAL that we have at the end of the first step of glycolysis reduce NAD+ to NADH, which is carrying for us high energy electrons and hydrogen that we'll send to the electron transport chain to get energy out of later. We can add that to our metabolic pathway for that next step. So now we have BPGA, or biphosphoglycerate. Let's look what happens to those molecules. The BPGA will phosphorylate ADP into ATP, and BPGA becomes PGA, or phosphoglycerate. And these ATP were made by substrate level phosphorylation. And let's add that to our flow chart here. So the BPGA phosphorylate ADP into ATP, becoming phosphoglycerate. The 2 phosphoglycerate then phosphorylate another 2 ADP into 2 more ATP, again by substrate level phosphorylation. And we end with 2 molecules of pyruvate. So let's finish this out. The two PGAs phosphorylate ADP into ATP, producing 2-pyruvate. So in this second stage, we've made 4 ATP. So we call this our energy return stage. We have a net profit of 2 ATP. We spin 2, we make 4. We also have these 2 NADH molecules that are carrying energy that we've yet to tap into. So let's summarize glycolysis. Again, it's anaerobic, it doesn't require oxygen, it takes place in the cytoplasm. We have an energy investment step of 2 ATP, but an energy return of 4 for a net yield or a profit of 2 ATP. We also produce 2 NADH molecules, which will go onto the electron transport chain, and we end with 2 pyruvate molecules, which will go onto the Krebs. So again, when we're looking at our flow chart, the products of glycolysis, besides this 4 ATP that we're after a profit of 2, the other 2 products, the NADH and the pyruvate, we're not done with. These pyruvates still have energy in them that we're going to tap into in the Krebs cycle, and the NADH have energy in them that we'll tap into in the electron transport chain. Finally, let's finish with a little accounting. Let's see, here's the overall reaction for respiration, and we've accomplished so far just glycolysis. So let's take into account what we have and have not done yet. And let me grab a pen here. So we use glucose. Check. We're done with that. We've yet to use oxygen, and we've yet to make carbon dioxide, and we've yet to make water. But we have made some ATP, but only 2. So we have approximately 34 ATP left to make. So where's the energy for those ATP? Well, hopefully you can see that we've yet to tap into these molecules, and so that's where we're going to go next. We obviously still have a long way to go to get through this whole process.
We know by now that glycolysis takes place in the cytoplasm and leads to the formation of 2 molecules of pyruvate per molecule of glucose. These pyruvate molecules are converted into acetyl coenzyme A, also known as acetyl-CoA, and this happens inside the mitochondrial matrix. Therefore, acetyl-CoA is the key metabolic intermediate that links glycolysis and the citric acid cycle. For clarity, we will not show the coenzyme portion of acetyl-CoA, and instead are simply representing it as the 2-carbon molecule acetate. So how does the pyruvate get into the mitochondrial matrix? This depends on a carrier protein that transports pyruvate into the mitochondrial matrix, where there it is oxidized to form acetyl-CoA by the enzyme pyruvate dehydrogenase. In this reaction, a molecule of carbon dioxide is generated as pyruvate is oxidized to acetyl-CoA, and a molecule of NAD+ is reduced to NADH. This last reaction is not shown for clarity. The citric acid cycle consists of the following 8 steps, and also harvests chemical energy, producing a total of 3 NADH molecules. In the first step of the citric acid cycle, the enzyme citrate synthase combines the 2-carbon acetyl group from acetyl-CoA with the 4-carbon molecule oxaloacetate. This generates the 6-carbon molecule citrate. In the second step, citrate is isomerized into isocitrate by a conitase. In the third step, isocitrate is oxidized by isocitrate dehydrogenase, generating the 5-carbon molecule alpha-ketoglutarate. In the process, 1 carbon dioxide molecule is released, and 1 NAD+ molecule is reduced to NADH. In the fourth step, alpha-ketoglutarate is decarboxylated and oxidized to produce another molecule of carbon dioxide by alpha-ketoglutarate dehydrogenase. The resulting molecule is combined with coenzyme A, forming succinyl-CoA. In addition, another molecule of NAD+ is reduced to form NADH. Note that in this diagram, we are showing the product of this reaction as succinate and are omitting the coenzyme A for clarity. In the fifth step, succinyl coenzyme A is converted into succinate by succinyl-CoA synthetase, releasing coenzyme A. This reaction also produces 1 molecule of GTP, another high-energy molecule, which is omitted from this diagram for clarity. In the sixth step, succinate is oxidized to fumarate by succinate dehydrogenase. Unlike all the other enzymes in the citric acid cycle, succinate dehydrogenase is embedded in the inner mitochondrial membrane and is also a component of the electron transport chain, in which it is also called complex 2. As complex 2 oxidizes succinate, it receives 2 electrons that it will ultimately pass on to coenzyme Q, which in turn will deliver its electrons to complex 3. Another interesting aspect of complex 2 is that it uses FAD rather than NAD+ to accept electrons. In the seventh step, fumarate is converted to malate by the enzyme fumarase. And finally, in the eighth step, the enzyme malate dehydrogenase oxidizes malate to reform oxaloacetate and reduces a molecule of NAD+ to NADH in the process. This regenerated oxaloacetate can then combine with another acetyl-CoA in the next cycle.
So we already know that if we start off with a glucose molecule, which is a 6-carbon molecule. 6-carbon molecule, that this essentially gets split in half by glycolysis, and we end up with 2 pyruvic acids, or 2 pyruvate molecules. So glycolysis literally splits this in half. It lyses the glucose, and we end up with 2 pyruvates, or pyruvic acids. Pyruvate. And these are 3-carbon molecules. 3-carbon molecules. There's obviously a lot of other stuff going on in the carbons. You saw it in the past, and you could look up their chemical structures on the internet or Wikipedia and see them in detail. But this is kind of the important thing, is that it was lysed, it was cut in half, and this is what happened in glycolysis. Glycolysis. And this happened in the absence of oxygen, or not necessarily. It can happen in the presence or in the absence of oxygen. It doesn't need oxygen. And we got a net payoff of 2 ATPs. 2 ATPs net. And I always say the net there because you remember, it used 2 ATPs in kind of that investment stage, and then it generated 4. So on a net basis, it generated 4, used 2, it gave us 2 ATPs, and it also produced 2 NADHs. 2 NADHs. That's what we got out of glycolysis. And just so you can visualize this a little bit better, let me draw a cell right here. Maybe I'll draw it down here. Let's say I have a cell. That's its outer membrane. Maybe its nucleus. We're dealing with the eukaryotic cell. Doesn't have to be the case. It has its DNA in its chromatin form all spread around like that. And then you have some mitochondria, and there's a reason why people call it the power centers of the cell. We'll look at that in a second. So there's a mitochondria. It has an outer membrane and an inner membrane, just like that. I'll do more detail on the structure of a mitochondria maybe later in this video, or maybe I'll do a whole video on them. That's another mitochondria right there. And then all of this fluidic space out here that's between the organelles, where in the organelles you can view them as parts of the cell that are, they do specific things. They do specific things, kind of like organs do specific things within our own bodies. So between all of the organelles you have just this fluidic space. This is just fluid of the cell, and that's called the cytoplasm. Cytoplasm. And that's where glycolysis occurs. So glycolysis occurs in the cytoplasm. Glycolysis. Now, we all know, in the overview video, we know what the next step is. It's the Krebs cycle, or the citric acid cycle. And that actually takes place in the inner membrane, or the inner space. The inner space of these mitochondria. Let me draw it a little bit bigger. Let me draw a mitochondria here. So if this is a mitochondria, it has an outer membrane. It has an inner membrane. If I have just one inner membrane, we call it a crista. If we have many, we call them cristae. These little convoluted inner membrane. Let me give it a label. I'll say they're cristae, the plural. And then it has 2 compartments, right? It's because it's divided by these 2 membranes. This compartment right here is called the outer compartment. This whole thing right there, that's the outer compartment. And then this inner compartment in here, This inner compartment in here is called the matrix. The inner compartment right there is called the matrix. Now, you have these pyruvates. They're not quite just ready for the Krebs cycle, but I guess, well, it's a good intro into how do you make them ready for the Krebs cycle? They actually get oxidized, and I'll just focus on one of these pyruvates. And we just have to remember that the pyruvate, that this happens twice for every molecule of glucose. So we have this kind of preparation step for the Krebs cycle. We call that pyruvate oxidation. Pyruvate oxidation. And essentially what it does is it cleaves one of these carbons off of the pyruvate, and so you end up with a 2-carbon compound. You don't have just 2 carbons, but its backbone of carbons is just 2 carbons, called acetyl-CoA. Acetyl-CoA. And if these names are confusing, because, what's acetyl coenzyme A? These are very bizarre. You could do a web search on them, but I'm just going to use the words right now because it'll keep things simple and we'll get the big picture. So it generates acetyl-CoA, which is this 2-carbon compound, and it also reduces some NAD plus to NADH. And this process right here is often given credit, or the Krebs cycle or the citric acid cycle gets credit for this step, but it's really a preparation step for the Krebs cycle. Now, once you have this 2-carbon chain acetyl-CoA right here, you are ready to jump into the Krebs cycle, this long talked about Krebs cycle, and you'll see in a second why it's called a cycle. The acetyl-CoA, and all of this is catalyzed by enzymes. And enzymes are just proteins that bring together the constituent things that need to react in the right way so that they do react. So catalyzed by enzymes, this acetyl-CoA merges with some oxaloacetic acid, very fancy word, but this is a 4-carbon molecule. This is a 4-carbon molecule right here. These two guys are kind of reacted together or merged together, depending on how you want to view it. Draw it like that. It's all catalyzed by enzymes. It's important, some tests will say, is this an enzyme catalyzed reaction? Yes, everything in the Krebs cycle is an enzyme catalyzed reaction, and they form citrate or citric acid. Citric acid, which is the same stuff in your lemonade or your orange juice. And this is a 6-carbon molecule, which makes sense. You have a 2-carbon and a 4-carbon. You get a 6-carbon molecule. And then the citric acid is then oxidized over a bunch of steps. And this is a huge simplification here, but it's just oxidized over a bunch of steps. Again, the carbons are cleaved off. Both 2 carbons are cleaved off of it to get back to oxaloacetic acid. And you might be saying, hey, when these carbons are cleaved off, like, when, let's say, this carbon is cleaved off, what happens to it? It becomes CO2. It gets put onto some oxygen and leaves the system. So this is where the oxygen or the carbons or the carbon dioxide actually gets formed. And similarly, when these carbons get cleaved off, it forms CO2. And actually, for every molecule of glucose, you have 6 carbons. When you do this whole process once, you're generating 3 molecules of carbon dioxide, but you're going to do it twice. You're going to have 6 carbon dioxides produced, which accounts for all of the carbons, right? You get rid of 3 carbons for every turn of this, well, 2 for every turn, but really for the steps after glycolysis, you get rid of 3 carbons, but you're going to do it for each of the pyruvates. So you're going to get rid of all 6 carbons, which will have to exhale eventually. But this cycle, it doesn't just generate carbons. The whole idea is to generate NADHs and FADH2s and ATPs. So, right here, and this is a huge simplification. I'll show you the detailed picture in a second. We'll reduce some NAD into NADH, NAD plus into NADH. We'll do it again. And of course, these are in separate steps. There's intermediate compounds. I'll show you those in a second. NAD plus, another NAD plus molecule will be reduced to NADH. Maybe we'll produce some ATP will turn into, or some ADP will turn into ATP. ADP turning into ATP. Maybe we have some, and not maybe, this is what happens. Some FAD gets, let me write it this way. Some FAD gets oxidized into FADH2. And the whole reason why we even pay attention to these You might think, hey, cellular respiration is all about ATP. Why do we even pay attention to these NADHs and these FADH2s that get produced as part of the process? The reason why we care is that these are the inputs into the electron transport chain. These get oxidized, or they lose their hydrogens in the electron transport chain, and that's where the bulk of the ATP is actually produced. And then maybe we'll have another NAD get reduced or gain in hydrogen. Right? Reduction is gaining an electron or gaining a hydrogen whose electron you can hog. NADH. And then we end up back at oxaloacetic acid, and we can perform the whole citric acid cycle over again. So now that we've written it all out, let's account for what we have. So depending on, let me draw some dividing lines so we know what's what. So this right here, everything to the left of that line right there is glycolysis. Glycolysis. We learned that already. And then most, especially introductory textbooks, will give the Krebs cycle credit for this pyruvate oxidation, but that's really a preparatory stage. The Krebs cycle is really formally this part where you start with acetyl-CoA, you merge it with oxaloacetic acid, and then you go and you form citric acid, and which essentially gets oxidized and produces all of these things that will need to either directly produce ATP or you do it indirectly in the electron transport chain. But let's account for everything that we have. Let's account for everything that we have so far. We already accounted for the glycolysis right there, 2 net ATPs, 2 NADHs. Now, in the citric acid cycle, or in the Krebs cycle, well, first we have our pyruvate oxidation that produced 1 NADH. But remember, if we want to say what are we producing for every glucose, this is what we produced for each of the pyruvates, right? This was from 1 NADH from just this pyruvate. But glycolysis produced 2 pyruvates. So everything after this we're going to multiply by 2 for every molecule of glucose. So I'll say so the pyruvate oxidation times 2 means that we got 2 NADHs, 2 NADHs right there. And then when we look at this side, the formal Krebs cycle, what do we get? We have how many NADHs? 1, 2, 3 NADHs. So 3 NADHs times 2, because we're going to perform this cycle for each of the pyruvates produced from glycolysis. So that gives us 6 NADHs. We have 1 ATP per turn of the cycle. This is going to happen twice, once for each pyruvic acid. So we get 2 ATPs. And then we have 1 FADH2, but we're going to do this cycle twice. This is per cycle, so times 2. We have 2 FADHs. Now, sometimes in a lot of books, these 2 NADHs, or per turn of the Krebs cycle, or per pyruvate, this 1 NADH, they'll give credit to the Krebs cycle for that. So sometimes instead of having this intermediate step, they'll just write 4 NADHs right here, and you're going to do it twice, once for each pyruvate. So they'll say 8 NADHs get produced from the Krebs cycle. But the reality is 6 from the Krebs cycle, 2 from the preparatory stage. Now, the interesting thing is we can now count whether we get to the 38 ATPs promised by cellular respiration. We've directly already produced for every molecule of glucose 2 ATPs and then 2 more ATPs. So we have 4 ATPs. 4 ATPs. How many NADHs do we have? 2, 4, and then 4 plus 6 is 10. We have 10 NADHs. And then we have 2 FADHs. 2 FADH2s. I think in the first video on cellular respiration I said FADH. It should be FADH2 just to be particular about things. And these, so you might say, hey, where's our 38 ATPs? We only have 4 ATPs right now. But these are actually the inputs in the electron transport chain. These molecules right here get oxidized in the electron transport chain. Every NADH in the electron transport chain produces 3 ATPs. So these 10 NADHs are going to produce 30 ATPs in the electron transport chain. And each FADH2, when it gets oxidized and gets turned back into FAD in the electron transport chain, will produce 2 ATPs. So 2 of them are going to produce 4 ATPs in the electron transport chain. So we now see we get 4 from just what we've done so far: glycolysis, the preparatory stage, and the Krebs or citric acid cycle. And then eventually these outputs from glycolysis and the citric acid cycle, when they get into the electron transport chain, are going to produce another 34. So 34 plus 4, it does get us to the promised 38 ATP that you would expect in a super efficient cell. This is kind of the theoretical maximum. In most cells, they really don't get quite there, but this is the good number to know if you're going to take the AP Bio test or in most introductory biology courses. There's one other point I want to make here. Everything we've talked about so far, this is carbohydrate metabolism, or sugar catabolism, we could call it. We're breaking down sugars to produce ATP. Glucose was our starting point. But animals, including us, we can catabolize other things. We can catabolize proteins. We can catabolize fats. I mean, if you have any fat on your body, you have energy. You're in theory, your body should be able to take that fat and you should be able to do things with that. You should be able to generate ATP. And the interesting thing, the reason why I bring it up here is obviously glycolysis is of no use to these things, although fats can be turned into glucose in the liver. But what the interesting thing is that the Krebs cycle is the entry point for these other catabolic mechanisms. Proteins can be broken down into amino acids, which can be broken down into acetyl-CoA. Fats can be turned into glucose, which actually could then go the whole cellular respiration. But the big picture here is acetyl-CoA is the general catabolic kind of intermediary that can then enter the Krebs cycle and generate ATP regardless of whether our fuel is carbohydrates, sugars, proteins, or fats. Now, we have a good sense of how everything works out right now, I think. Now I'm going to show you kind of a diagram that you might see in your biology textbook, or I'll actually show you the actual diagram from Wikipedia. And I just want to show you, this looks very daunting and very confusing, and I think that's why many of us have trouble with cellular respiration initially, because there's just so much information. It's hard to process what's important. But I want to just highlight the important steps here, just so you see it's the same thing that we talked about. From glycolysis, you produce 2 pyruvates. That's the pyruvate right there. They actually showed some molecular structure. This is the pyruvate oxidation step that I talked about, the preparatory step. And you see we produce a carbon dioxide, and we reduce an NAD plus into NADH. Then we're ready to enter the Krebs cycle. The acetyl-CoA and the oxaloacetate, or oxaloacetic acid, they are reacted together to create citric acid. They've actually drawn the molecule there. And then the citric acid is oxidized through the Krebs cycle right there. All of these steps, each of these steps are facilitated by enzymes, and it gets oxidized. But I want to highlight the interesting parts. Here we have an NAD get reduced to NADH. We have another NAD get reduced to NADH. And then over here, another NAD get reduced to NADH. So far, if you include the preparatory step, we've had 4 NADHs formed, 3 directly from the Krebs cycle. That's just what I told you. Now we have, in this diagram, they say GDP or GTP gets formed from GDP. The GTP is just guanosine triphosphate. It's another purine that can be a source of energy. But then that later can be used to form an ATP. So this is just the way they happen to draw it, but this is the actual ATP that I drew in the diagram on the top. And then they have this Q group, and I won't go into it, and then it gets reduced over here. It gets those 2 hydrogens, but that essentially ends up reducing the FADH2s. So this is where the FADH2 gets produced. So as promised, we produced for each pyruvate that inputted, remember, so we're going to do it twice. For each pyruvate, we produced 1, 2, 3, 4 NADHs. We produced 1 ATP and 1 FADH2. And that's exactly what we saw up here.
Krebs cycle is also known as the tricarboxylic acid cycle, and it's a biochemical pathway that is used to generate energy through the oxidation of acetyl-CoA. It is also used for the synthesis of NADH and for the production of amino acids. Krebs cycle takes place in the mitochondria of the eukaryotes. The first step involved is actually a preparatory step which begins with pyruvate. Pyruvate is derived through the glycolysis of glucose, which is a 6-carbon compound, and it is split into 2 molecules of pyruvate, which is a 3-carbon compound. The next step includes the oxidation of pyruvate into acetyl-CoA by the enzyme pyruvate dehydrogenase complex. And in this reaction, a molecule of carbon dioxide and a molecule of NADH is generated. The acetyl-CoA is a 2-carbon compound. In the next step, the acetyl-CoA combines with oxaloacetate, which is a 4-carbon compound, to form citrate. And hence the resulting molecule is a 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase. In the next step, the citrate is isomerized into isocitrate by the enzyme aconitase. Next, the isocitrate is oxidized into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. And in this reaction, a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is generated. Hence, the alpha-ketoglutarate is a 5-carbon compound since one molecule of carbon is lost into carbon dioxide. Next, the alpha-ketoglutarate is converted into succinyl-CoA by the enzyme alpha-ketoglutarate dehydrogenase. And in this reaction also, a molecule of NAD is reduced to NADH. And a molecule of carbon dioxide is released. Hence, the succinyl-CoA is a 4-carbon compound. In the next step, the succinyl-CoA is converted into succinate by the enzyme succinyl-CoA synthase. In this reaction, a molecule of GTP is generated. Next, succinate is converted into fumarate by the enzyme succinate dehydrogenase. In this reaction, a molecule of QH2 is generated, which is used for the production of FADH2. Fumarate is then converted into malate by the enzyme fumarase. And in the last step, the malate is converted into oxaloacetate by the enzyme malate dehydrogenase. And in this reaction also, NAD is reduced to NADH. Now let's look at the results of the Krebs cycle. Through each cycle of the Krebs cycle, following products are generated: 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of carbon dioxide. Now since glucose is split into 2 pyruvate compounds, for each molecule of glucose, this cycle runs twice. Hence the products formed are 6 NADH, 2 FADH2, 2 GTP, and 4 carbon dioxide. All the NADH and FADH are next fed to the electron transport chain for the generation of ATP.
Welcome back for part 2 on our series on cellular respiration. In this video, we'll focus on the details of the Krebs cycle. At the end of our last video, we had finished glycolysis and we had taken an accounting. We had made 2 of our 36 ATP, but we hadn't gotten much else done. We'd broken glucose in half to pyruvate, and we had made 2 NADH. They're going to go to the electron transport chain. That'll be video 3. But these 2 pyruvate, they head to the mitochondria for the Krebs cycle. The pyruvic acid, or pyruvate, enters the mitochondria. But before we can start the Krebs cycle, we need to prepare the pyruvate in what's called the prep step. A coenzyme called coenzyme A binds to the pyruvate, and NADH is reduced to make NADH. In doing so, a carbon dioxide is kicked out. That's the first of those we've produced. It'll be released as a waste product of respiration. The NADH will head off to the electron transport chain, and what's left is the acetyl-CoA, which will enter into the Krebs cycle. Let's draw this in a useful way. Here's the first, or the preparatory step. We're going to prepare for the Krebs cycle. It's the molecule acetyl-CoA, a 2-carbon molecule that enters the Krebs cycle. So 1 carbon is already taken care of. Now remember, we made 2 pyruvate in glycolysis, so we have to double all of our numbers. As acetyl-CoA enters the Krebs cycle, it reacts with oxaloacetate to to make citrate, thus the citric acid cycle, another name for the Krebs cycle. So we can draw that like this. Oxaloacetate picks up acetyl-CoA, the coenzyme A gets kicked out, and we have citrate. Now, what we have after this is a series of transfers, or a series of reactions, where we're going to convert the citrate. It's a cycle, so we're going to get back to oxaloacetate. And in the meantime, let's look at what we produce. In the first conversion, citrate is converted into an intermediate with the input of high energy electrons, and kicks out high energy electrons to reduce NAD+ into NADH. In doing so, each citrate loses a carbon, so we make 2 carbon dioxides because we have 2 citrates. I'm not going to require you to learn the name of this intermediate, and I'll show you why in just a moment. But that intermediate is then converted into a second intermediate, and we have the same process. We reduce 2 more NADH+ and 2 more to make 2 more NADH, and we kick out 2 more carbon dioxides. Now, we need to double the numbers here for our intermediates because we have 2 of everything, so I'm going to grab a pen. I should have a 2 in front of that and a 2 in front of this. Now let's not forget the point of what we're trying to do. We're trying to make ATP, and so far we've not made any. In this next change, intermediate 2 becomes intermediate 3, and in doing so we do make our first 2 ATP. 2 intermediates to make 2 ATP, and we do this by substrate level phosphorylation. We have 2 more reactions to go. We go to 1 more intermediate, and this time we reduce a molecule called FAD+. Now FAD is a lot like NAD in that it's an electron carrier. It's going to pick up hydrogen and high energy electrons. And finally, we need to convert back to oxaloacetate, and doing that we once again reduce 2 NAD+ to 2 NADH. Now this looks like a mess, but it could be a lot worse. Here's a diagram that I found in a textbook that shows the citric acid cycle, and you can see it could be a lot worse in terms of what you're required to know for our course versus what you might need to know for some other biochemistry course or such. You can see all the names of all the intermediate molecules, which you don't need to know, but you do need to know the first that picks the oxaloacetate that picks up acetyl-CoA, and the first product made, citrate. And the reason I show it to you like this is I do want you to understand that it's a series of reactions of intermediate to intermediate to intermediate, and that it's not just a kind of a simple thing. And like I said, you don't need to necessarily know the names of these intermediates, or you don't need to know them at all. But I do think it's important to understand that it happens in stages, that we're getting this energy out of these molecules, these high energy bonds that are now in citrate, that were in acetyl-CoA, that we're tapping into that energy and getting it in little pieces, and then we still have to get to that energy, because we've only made 2 ATP. Now we can kind of summarize this in a little simpler form. This may be easier. If we look at it in sum total, in the prep step we're making 2 carbon dioxides, waste product, and 2 NADH, which are going to go to the electron transport chain. In this Krebs cycle we produce 2 ATP by substrate level phosphorylation. We kick out 4 carbon dioxide, so 2 and 4, that's 6, that's all of them. We produce a total of 6 more NADH and 2 FADH2. All these electron carriers here, here, and here are going to go to the electron transport chain. There's still energy in those molecules. So all we have left to do is summarize and give an accounting. So, the Krebs cycle. Pyruvate is prepared for the Krebs cycle as converted to acetyl-CoA. 2 carbon dioxides and 2 NADH are produced in this prep step. The acetyl-CoA is picked up by oxaloacetate and converted to citrate, and then progressing through a series of intermediates, 4 more carbon dioxides, 6 NADH, and 2 FADH2s, and 2 ATP are made. ATP made by substrate level phosphorylation. And for our accounting, here we add the prep step and the Krebs cycle to this page, and let's see where we're at now. Have we used oxygen yet? Have we used oxygen? No. Have we made 6 carbon dioxides? Yes, we made 2 in the prep step and 4 in the Krebs cycle. Have we made any water? No. How about energy? Well, in the Krebs cycle we make 2 ATP. So that's 2 more we can subtract from our total, so we still have about 32 ATP to make. But we have nothing left to break down. We've taken a 6-carbon molecule, glucose, and made 6 carbon dioxides. But what do we have left to do? What can we still tap into? Well, if you can see along the way, we've been picking up NADHs, 10 of them, 2 from glycolysis, 2 from the prep step. Here we go. Oops, let me turn my pen over. 2 from glycolysis, 2 from the prep step, 6 from the Krebs cycle, plus these 2 FADH2. All of these molecules are going to be sent to the electron transport chain for electron transport phosphorylation. There's still a lot of energy that we have to tap into in these molecules. In fact, the majority of that energy. And notice we've yet to use oxygen and we still haven't made water, so there's a lot more to do.